Photo shows a shark swimming toward the camera. Figure 36. 1. This shark uses its senses of sight, vibration, lateral line system, and smell to hunt, but it also relies on its ability to sense the electric fields of prey, a sense not present in most land animals. Credit, modification of work by Hermanus Backpackers Hostel, South Africa, chapter outline 36. 1. Sensory processes 36. 2. Somatosensation 36. 3. Taste and smell 36. 4. Hearing and vestibular sensation 36. 5. Vision. In more advanced animals, the senses are constantly at work, making the animal aware of stimuli, such as light or sound, or the presence of a chemical substance in the external environment, and monitoring information about the organism's internal environment. All bilaterally symmetric animals have a sensory system, and the development of any species' sensory system has been driven by natural selection. Thus, sensory systems differ among species according to the demands of their environments. The shark, unlike most fish predators, is electrosensitive. That is, sensitive to electrical fields produced by other animals in its environment. While it is helpful to this underwater predator, electrosensitivity is a sense not found in most land animals. Learning objectives. By the end of this section, you will be able to do the following. Identify the general and special senses in humans, describe 3 important steps in sensory perception, explain the concept of just noticeable difference in sensory perception senses, provide information about the body and its environment. Humans have 5 special senses: olfaction, smell, gustation, taste, equilibrium, balance and body position, vision, and hearing. Additionally, we possess general senses, also called somatosensation, which respond to stimuli like temperature, pain, pressure, and vibration. Vestibular sensation, which is an organism's sense of spatial orientation and balance, proprioception, position of bones, joints, and muscles, and the sense of limb position that is used to track kinesthesia, limb movement, are part of somatosensation. Although the sensory systems associated with these senses are very different, they all share a common function: to convert a stimulus, such as light or sound or the position of the body, into an electrical signal in the nervous system. This process is called sensory transduction. There are 2 broad types of cellular systems that perform sensory transduction. In one, a neuron works with a sensory receptor, a cell or cell process that is specialized to engage with and detect a specific stimulus. Stimulation of the sensory receptor activates the associated afferent neuron, which carries information about the stimulus to the central nervous system. In the second type of sensory transduction, a sensory nerve ending responds to a stimulus in the internal or external environment. This neuron constitutes the sensory receptor. Free nerve endings can be stimulated by several different stimuli, thus showing little receptor specificity. For example, pain receptors in your gums and teeth may be stimulated by temperature changes, chemical stimulation, or pressure. Reception. The first step in sensation is reception, which is the activation of sensory receptors by stimuli such as mechanical stimuli, being bent or squished, for example, chemicals or temperature. The receptor can then respond to the stimuli. The region in space in which a given sensory receptor can respond to a stimulus, be it far away or in contact with the body, is that receptor's receptive field. Think for a moment about the differences in receptive fields for the different senses. For the sense of touch, a stimulus must come into contact with the body. For the sense of hearing, a stimulus can be a moderate distance away. Some baleen whale sounds can propagate for many kilometers. For vision, a stimulus can be very far away. For example, the visual system perceives light from stars at enormous distances. Transduction. The most fundamental function of a sensory system is the translation of a sensory signal to an electrical signal in the nervous system. This takes place at the sensory receptor, and the change in electrical potential that is produced is called the receptor potential. How is sensory input, such as pressure on the skin, changed to a receptor potential? In this example, a type of receptor called a mechanoreceptor, as shown in figure 36. 2. Possesses specialized membranes that respond to pressure. Disturbance of these dendrites by compressing them or bending them opens gated ion channels in the plasma membrane of the sensory neuron, changing its electrical potential. Recall that in the nervous system, a positive change of a neuron's electrical potential, also called the membrane potential, depolarizes the neuron. Receptor potentials are graded potentials. The magnitude of these graded receptor potentials varies with the strength of the stimulus. If the magnitude of depolarization is sufficient, that is, if membrane potential reaches a threshold, the neuron will fire an action potential. In most cases, the correct stimulus impinging on a sensory receptor will drive membrane potential in a positive direction. Although for some receptors, such as those in the visual system, this is not always the case. Illustration A shows a closed gated ion channel embedded in the plasma membrane. A hair-like tether connects the channel to the extracellular matrix outside the cell, and another tether connects the channel to the inner cytoskeleton. When the extracellular matrix is deflected, the tether tugs on the gated ion channel, pulling it open. Ions may now enter or exit the cell. Illustration B shows stereocilia, hair-like projections on outer hair cells that are attached to the tectorial membrane of the inner ear. The outer hair cells are connected to the cochlear nerve. Figure 36.2. A. Mechanosensitive ion channels are gated ion channels that respond to mechanical deformation of the plasma membrane. A mechanosensitive channel is connected to the plasma membrane and the cytoskeleton by hair-like tethers. When pressure causes the extracellular matrix to move, the channel opens, allowing ions to enter or exit the cell. B. Stereocilia in the human ear are connected to mechanosensitive ion channels. When a sound causes the stereocilia to move, mechanosensitive ion channels transduce the signal to the cochlear nerve. Sensory receptors for different senses are very different from each other, and they are specialized according to the type of stimulus they sense. They have receptor specificity. For example, touch receptors, light receptors, and sound receptors are each activated by different stimuli. Touch receptors are not sensitive to light or sound. They are sensitive only to touch or pressure. However, stimuli may be combined at higher levels in the brain, as happens with olfaction, contributing to our sense of taste. Encoding and transmission of sensory information. 4 aspects of sensory information are encoded by sensory systems: the type of stimulus, the location of the stimulus in the receptive field, the duration of the stimulus, and the relative intensity of the stimulus. Thus, action potentials transmitted over a sensory receptor's afferent axons encode one type of stimulus, and this segregation of the senses is preserved in other sensory circuits. For example, auditory receptors transmit signals over their own dedicated system, and electrical activity in the axons of the auditory receptors will be interpreted by the brain as an auditory stimulus, a sound. The intensity of a stimulus is often encoded in the rate of action potentials produced by the sensory receptor. Thus, an intense stimulus will produce a more rapid train of action potentials, and reducing the stimulus will likewise slow the rate of production of action potentials. A second way in which intensity is encoded is by the number of receptors activated. An intense stimulus might initiate action potentials in a large number of adjacent receptors, while a less intense stimulus might stimulate fewer receptors. Integration of sensory information begins as soon as the information is received in the CNS, and the brain will further process incoming signals. Perception is an individual's interpretation of a sensation. Although perception relies on the activation of sensory receptors, perception happens not at the level of the sensory receptor, but at higher levels in the nervous system, in the brain. The brain distinguishes sensory stimuli through a sensory pathway. Action potentials from sensory receptors travel along neurons that are dedicated to a particular stimulus. These neurons are dedicated to that particular stimulus and synapse with particular neurons in the brain or spinal cord. All sensory signals, except those from the olfactory system, are transmitted through the central nervous system and are routed to the thalamus and to the appropriate region of the cortex. Recall that the thalamus is a structure in the forebrain that serves as a clearinghouse and relay station for sensory, as well as motor signals. When the sensory signal exits the thalamus, it is conducted to the specific area of the cortex. Figure 36. 3. Dedicated to processing that particular sense. How are neural signals interpreted? Interpretation of sensory signals between individuals of the same species is largely similar, owing to the inherited similarity of their nervous systems. However, there are some individual differences. A good example of this is individual tolerances to a painful stimulus, such as dental pain, which certainly differ. Illustration A shows side view of a human brain. The thalamus is in the inner, middle part. Illustration B shows the location of sensory processing regions in the brain. The visual processing region is at the back of the brain, the auditory processing region is in the middle of the brain, and the somatosensory processing region is located in a sliver-like region in the upper part of the brain and extending halfway down. Figure 36. 3. In humans, with the exception of olfaction, all sensory signals are routed from the A thalamus to B final processing regions in the cortex of the brain. Credit B. Modification of work by Polina Tishina. Scientific method connection, just noticeable difference. It is easy to differentiate between a 1-pound bag of rice and a 2-pound bag of rice. There is a 1-pound difference, and one bag is twice as heavy as the other. However, would it be as easy to differentiate between a 20 and a 21-pound bag? Question. What is the smallest detectable weight difference between a 1-pound bag of rice and a larger bag? What is the smallest detectable difference between a 20-pound bag and a larger bag? In both cases, at what weights are the differences detected? This smallest detectable difference in stimuli is known as the just noticeable difference, JND. Background. Research background literature on JND and on Weber's law. A description of a proposed mathematical relationship between the overall magnitude of the stimulus and the JND. You will be testing JND of different weights of rice in bags. Choose a convenient increment that is to be stepped through while testing. For example, you could choose 10% increments between 1 and 2 pounds, 1, 1, 1, 2, 1, 3, 1, 4, and so on, or 20% increments, 1, 2, 1, 4, 1, 6, and 1, 8. Hypothesis. Develop a hypothesis about JND in terms of percentage of the whole weight being tested, such as the JND between the 2 small bags and between the 2 large bags is proportionally the same, or is not proportionally the same. So, for the first hypothesis, if the JND between the 1-pound bag and a larger bag is 0.2 pounds, that is, 20%, 1 0-pound feels the same as 1 pounds, but 1 0-pound feels less than 1 2 pounds. Then the JND between the 20-pound bag and a larger bag will also be 20%. So, 20 pounds feels the same as 22 pounds or 23 pounds, but 20 pounds feels less than 24 pounds. Test the hypothesis. Enlist 24 participants and split them into 2 groups of 12. To set up the demonstration, assuming a 10% increment was selected, have the first group be the 1-pound group. As a counterbalancing measure against a systematic error, however, 6 of the first group will compare 1 pound to 2 pounds and step down in weight 0 to 20, 10 to 19, and so on, while the other 6 will step up 10 to 11, 10 to 12, and so on. Apply the same principle to the 20-pound group, 20 to 40, 20 to 38, and so on, and 20 to 22, 20 to 24, and so on. Given the large difference between 20 and 40 pounds, you may wish to use 30 pounds as your larger weight. In any case, use 2 weights that are easily detectable as different. Record the observations. Record the data in a table similar to the table below. For the 1-pound and 20-pound groups, base weights, record a plus sign, plus, for each participant that detects a difference between the base weight and the step weight. Record a minus sign, for each participant that finds no difference. If 1/10 steps were not used, then replace the steps in the step weight columns with the step you are using. Results of JND testing, plus equals difference. Equals no difference, step weight 1 pound 20 pounds, step weight 1 122 1 224 1, 326 1, 428 1, 531 632 1 734 1, 836 1, 938 2, 040 table 36. 1. Analyze the data, report the results. What step weight did all participants find to be equal with 1 pound base weight? What about the 20-pound group? Draw a conclusion. Did the data support the hypothesis? Are the final weights proportionally the same? If not, why not? Do the findings adhere to Weber's law? Weber's law states that the concept that a just noticeable difference in a stimulus is proportional to the magnitude of the original stimulus. Learning objectives. By the end of this section, you will be able to do the following. Describe 4 important mechanoreceptors in human skin. Describe the topographical distribution of somatosensory receptors between glabrous and hairy skin. Explain why the perception of pain is subjective. Somatosensation is a mixed sensory category and includes all sensation received from the skin and mucous membranes, as well from as the limbs and joints. Somatosensation is also known as tactile sense, or more familiarly, as the sense of touch. Somatosensation occurs all over the exterior of the body and at some interior locations as well. A variety of receptor types embedded in the skin, mucous membranes, muscles, joints, internal organs, and cardiovascular system play a role. Recall that the epidermis is the outermost layer of skin in mammals. It is relatively thin, is composed of keratin-filled cells, and has no blood supply. The epidermis serves as a barrier to water and to invasion by pathogens. Below this, the much thicker dermis contains blood vessels, sweat glands, hair follicles, lymph vessels, and lipid-secreting sebaceous glands, figure 36. 4. Below the epidermis and dermis is the subcutaneous tissue, or hypodermis, the fatty layer that contains blood vessels, connective tissue, and the axons of sensory neurons. The hypodermis, which holds about 50% of the body's fat, attaches the dermis to the bone and muscle and supplies nerves and blood vessels to the dermis. Illustration shows a cross-section of mammalian skin. The outer epidermis is a thin layer, smooth on the outside. The middle dermis is much thicker than the epidermis. Blood, nerve, and lymph vessels run along the bottom of it, and smaller capillaries and nerve endings extend to the upper part. One nerve ends in a receptor. Sweat glands extend from the dermis into the epidermis. Hair follicles extend from the base of the dermis to the upper part where they are joined by oil glands. Hairs extend from the follicles through the epidermis and out of the skin. The hypodermis is a fatty layer beneath the dermis, below the blood vessels and bases of the hair follicles. Figure 36. 4 mammalian skin has 3 layers: an epidermis, a dermis, and a hypodermis. Credit: modification of work by Don Bliss, National Cancer Institute. Somatosensory receptors. Somatosensory receptors are classified into 4 categories: mechanoreceptors, thermoreceptors, pain receptors, nociceptors, and chemoreceptors. These categories are based on the nature of stimuli each receptor class transduces. What is commonly referred to as touch involves more than one kind of stimulus and more than one kind of receptor. Mechanoreceptors in the skin are described as encapsulated, that is, surrounded by a capsule, or unencapsulated, a group that includes free nerve endings. A free nerve ending, as its name implies, is an unencapsulated dendrite of a sensory neuron. Free nerve endings are the most common nerve endings in skin, and they extend into the middle of the epidermis. Free nerve endings are sensitive to painful stimuli, to hot and cold, and to light touch. They are slow to adjust to a stimulus and so are less sensitive to abrupt changes in stimulation. There are 3 classes of mechanoreceptors: tactile, proprioceptors, and baroreceptors. Mechanoreceptors sense stimuli due to physical deformation of their plasma membranes. They contain mechanically gated ion channels whose gates open or close in response to pressure, touch, stretching, and sound. There are 4 primary tactile mechanoreceptors in human skin: Merkel's discs, Meissner's corpuscles, Ruffini endings, and Pacinian corpuscles. 2 are located toward the surface of the skin, and 2 are located deeper. A 5th type of mechanoreceptor, Krause end bulbs, are found only in specialized regions. Merkel's discs, shown in figure 36. 5 are found in the upper layers of skin near the base of the epidermis, both in skin that has hair and on glabrous skin, that is, the hairless skin found on the palms and fingers, the soles of the feet, and the lips of humans and other primates. Merkel's discs are densely distributed in the fingertips and lips. They are slow-adapting, unencapsulated nerve endings, and they respond to light touch. Light touch, also known as discriminative touch, is a light pressure that allows the location of a stimulus to be pinpointed. The receptive fields of Merkel's discs are small with well-defined borders. That makes them finely sensitive to edges, and they come into use in tasks such as typing on a keyboard. Visual connection illustration shows the location of various mechanoreceptors in a cross-section of the epidermis and dermis. A nerve runs along the middle of the dermis, and all the mechanoreceptors are connected to it. Ruffini endings, Merkel's discs, and Meissner's corpuscles are all located in the upper dermis above the nerve. Ruffini endings are bulbous, horizontal mechanoreceptors located in the middle of the upper dermis. Meissner's corpuscles are bulbous, vertical mechanoreceptors that touch the bottom of the epidermis. Merkel's discs have finger-like projections that also touch the bottom of the epidermis. The roundish Krause end bulbs are also encapsulated within the dermis. The last type of mechanoreceptor, Pacini corpuscles, are oval mechanoreceptors located in the lower dermis. Figure 36. 4 of the primary mechanoreceptors in human skin are shown. Merkel's discs, which are unencapsulated, respond to light touch. Meissner's corpuscles, Ruffini endings, and Pacinian corpuscles are all encapsulated. Meissner's corpuscles respond to touch and low-frequency vibration. Ruffini endings detect stretch, deformation within joints, and warmth. Pacinian corpuscles detect transient pressure and high-frequency vibration. Krause end bulbs are encapsulated thermoreceptors that detect cold. Which of the following statements about mechanoreceptors is false? Pacinian corpuscles are found in both glabrous and hairy skin. Merkel's discs are abundant on the fingertips and lips. Ruffini endings are encapsulated mechanoreceptors. Meissner's corpuscles extend into the lower dermis. Meissner's corpuscles, shown in Figure 36. 6, also known as tactile corpuscles, are found in the upper dermis, but they project into the epidermis. They, too, are found primarily in the glabrous skin on the fingertips and eyelids. They respond to fine touch and pressure, but they also respond to low-frequency vibration or flutter. They are rapidly adapting, fluid-filled, encapsulated neurons with small, well-defined borders and are responsive to fine details. Like Merkel's discs, Meissner's corpuscles are not as plentiful in the palms as they are in the fingertips. Micrograph shows the epidermis, which stains dark pink, and the dermis, which stains light pink. Finger-like projections of epidermis extend into the dermis. Between two of these finger-like projections is an oval Meissner corpuscle about 10 microns across and 20 microns long. Figure 36. 6 Meissner corpuscles in the fingertips, such as the one viewed here using bright field light microscopy, allow for touch discrimination of fine detail. Credit: modification of work by W. Bensmith/Wikimedia Commons. Scale bar data from Matt Russell deeper in the epidermis, near the base, are Ruffini endings, which are also known as bulbous corpuscles. They are found in both glabrous and hairy skin. These are slow-adapting, encapsulated mechanoreceptors that detect skin stretch and deformations within joints, so they provide valuable feedback for gripping objects and controlling finger position and movement. Thus, they also contribute to proprioception and kinesthesia. Ruffini endings also detect warmth. Note that these warmth detectors are situated deeper in the skin than are the cold detectors. It is not surprising, then, that humans detect cold stimuli before they detect warm stimuli. Pacinian corpuscles, seen in Figure 36-7, are located deep in the dermis of both glabrous and hairy skin and are structurally similar to Meissner's corpuscles. They are found in the bone periosteum, joint capsules, pancreas and other viscera, breast, and genitals. They are rapidly adapting mechanoreceptors that sense deep transient, but not prolonged, pressure and high-frequency vibration. Pacinian receptors detect pressure and vibration by being compressed, stimulating their internal dendrites. There are fewer Pacinian corpuscles and Ruffini endings in skin than there are Merkel's discs and Meissner's corpuscles. Micrograph shows 3 Pacinian corpuscles embedded in the dermis. The corpuscles are round and about 1-4 millimeters across and have rings, like a tree stump. Figure 36. 7 Pacinian corpuscles, such as these visualized using bright field light microscopy, detect pressure, touch, and high-frequency vibration. Credit: Modification of work by Ed Uthman. Scale bar data from Matt Russell. In proprioception, proprioceptive and kinesthetic signals travel through myelinated afferent neurons running from the spinal cord to the medulla. Neurons are not physically connected, but communicate via neurotransmitters secreted into synapses or gaps between communicating neurons. Once in the medulla, the neurons continue carrying the signals to the thalamus. Muscle spindles are stretch receptors that detect the amount of stretch or lengthening of muscles. Related to these are Golgi tendon organs, which are tension receptors that detect the force of muscle contraction. Proprioceptive and kinesthetic signals come from limbs. Unconscious proprioceptive signals run from the spinal cord to the cerebellum, the brain region that coordinates muscle contraction, rather than to the thalamus, like most other sensory information. Baroreceptors detect pressure changes in an organ. They are found in the walls of the carotid artery and the aorta where they monitor blood pressure, and in the lungs where they detect the degree of lung expansion. Stretch receptors are found at various sites in the digestive and urinary systems. In addition to these two types of deeper receptors, there are also rapidly adapting hair receptors, which are found on nerve endings that wrap around the base of hair follicles. There are a few types of hair receptors that detect slow and rapid hair movement, and they differ in their sensitivity to movement. Some hair receptors also detect skin deflection, and certain rapidly adapting hair receptors allow detection of stimuli that have not yet touched the skin. Integration of signals from mechanoreceptors, the configuration of the different types of receptors working in concert in human skin, results in a very refined sense of touch. The nociceptive receptors, those that detect pain, are located near the surface. Small, finely calibrated mechanoreceptors, Merkel's discs and Meissner's corpuscles, are located in the upper layers and can precisely localize even gentle touch. The large mechanoreceptors, Pacinian corpuscles and Ruffini endings, are located in the lower layers and respond to deeper touch. Consider that the deep pressure that reaches those deeper receptors would not need to be finely localized. Both the upper and lower layers of the skin hold rapidly and slowly adapting receptors. Both primary somatosensory cortex and secondary cortical areas are responsible for processing the complex picture of stimuli transmitted from the interplay of mechanoreceptors. Density of mechanoreceptors, the distribution of touch receptors in human skin, is not consistent over the body. In humans, touch receptors are less dense in skin covered with any type of hair, such as the arms, legs, torso, and face. Touch receptors are denser in glabrous skin, the type found on human fingertips and lips, for example, which is typically more sensitive and is thicker than hairy skin, 4 to 5 versus 2 to 3. How is receptor density estimated in a human subject? The relative density of pressure receptors in different locations on the body can be demonstrated experimentally using a two-point discrimination test. In this demonstration, two sharp points, such as two thumbtacks, are brought into contact with the subject's skin, though not hard enough to cause pain or break the skin. The subject reports if they feel 1 point or 2 points. If the 2 points are felt as 1 point, it can be inferred that the 2 points are both in the receptive field of a single sensory receptor. If 2 points are felt as 2 separate points, each is in the receptive field of 2 separate sensory receptors. The points could then be moved closer and retested until the subject reports feeling only 1 point, and the size of the receptive field of a single receptor could be estimated from that distance. Thermoreception, in addition to Krause end bulbs that detect cold and Ruffini endings that detect warmth, there are different types of cold receptors on some free nerve endings. Thermoreceptors, located in the dermis, skeletal muscles, liver, and hypothalamus, that are activated by different temperatures. Their pathways into the brain run from the spinal cord through the thalamus to the primary somatosensory cortex. Warmth and cold information from the face travels through one of the cranial nerves to the brain. You know from experience that a tolerably cold or hot stimulus can quickly progress to a much more intense stimulus that is no longer tolerable. Any stimulus that is too intense can be perceived as pain because temperature sensations are conducted along the same pathways that carry pain sensations. Pain is the name given to nociception, which is the neural processing of injurious stimuli in response to tissue damage. Pain is caused by true sources of injury, such as contact with a heat source that causes a thermal burn or contact with a corrosive chemical. But pain also can be caused by harmless stimuli that mimic the action of damaging stimuli, such as contact with capsaicins, the compounds that cause peppers to taste hot and which are used in self-defense pepper sprays and certain topical medications. Peppers taste hot because the protein receptors that bind capsaicin open the same calcium channels that are activated by warm receptors. Nociception starts at the sensory receptors, but pain, inasmuch as it is the perception of nociception, does not start until it is communicated to the brain. There are several nociceptive pathways to and through the brain. Most axons carrying nociceptive information into the brain, from the spinal cord project to the thalamus, as do other sensory neurons, and the neural signal undergoes final processing in the primary somatosensory cortex. Interestingly, one nociceptive pathway projects not to the thalamus, but directly to the hypothalamus in the forebrain, which modulates the cardiovascular and neuroendocrine functions of the autonomic nervous system. Recall that threatening or painful stimuli stimulate the sympathetic branch of the visceral sensory system, readying a fight-or-flight response. Link to learning view this video that animates the 5 phases of nociceptive pain. Learning objectives. By the end of this section, you will be able to do the following. Explain in what way smell and taste stimuli differ from other sensory stimuli. Identify the 5 primary tastes that can be distinguished by humans. Explain in anatomical terms why a dog's sense of smell is more acute than a human's. Taste, also called gustation, and smell, also called olfaction, are the most interconnected senses in that both involve molecules of the stimulus entering the body and bonding to receptors. Smell lets an animal sense the presence of food or other animals, whether potential mates, predators, or prey, or other chemicals in the environment that can impact their survival. Similarly, the sense of taste allows animals to discriminate between types of foods. While the value of a sense of smell is obvious, what is the value of a sense of taste? Different tasting foods have different attributes, both helpful and harmful. For example, sweet-tasting substances tend to be highly caloric, which could be necessary for survival in lean times. Bitterness is associated with toxicity, and sourness is associated with spoiled food. Salty foods are valuable in maintaining homeostasis by helping the body retain water and by providing ions necessary for cells to function. Tastes and odors. Both taste and odor stimuli are molecules taken in from the environment. The primary tastes detected by humans are sweet, sour, bitter, salty, and umami. The first 4 tastes need little explanation. The identification of umami as a fundamental taste occurred fairly recently. It was identified in 1908 by Japanese scientist Kikini Ikeda while he worked with seaweed broth. But it was not widely accepted as a taste that could be physiologically distinguished until many years later. The taste of umami, also known as savoriness, is attributable to the taste of the amino acid L-glutamate. In fact, monosodium glutamate, or MSG, is often used in cooking to enhance the savory taste of certain foods. What is the adaptive value of being able to distinguish umami? Savory substances tend to be high in protein. All odors that we perceive are molecules in the air we breathe. If a substance does not release molecules into the air from its surface, it has no smell. And if a human or other animal does not have a receptor that recognizes a specific molecule, then that molecule has no smell. Humans have about 350 olfactory receptor subtypes that work in various combinations to allow us to sense about 10,000 different odors. Compare that to mice, for example, which have about 1,300 olfactory receptor types and therefore probably sense more odors. Both odors and tastes involve molecules that stimulate specific chemoreceptors. Although humans commonly distinguish taste as one sense and smell as another, they work together to create the perception of flavor. A person's perception of flavor is reduced if they have congested nasal passages. Reception and transduction. Odorants, odor molecules, enter the nose and dissolve in the olfactory epithelium, the mucosa at the back of the nasal cavity, as illustrated in Figure 36. 8. The olfactory epithelium is a collection of specialized olfactory receptors in the back of the nasal cavity that spans an area about 5 cm2 in humans. Recall that sensory cells are neurons. An olfactory receptor, which is a dendrite of a specialized neuron, responds when it binds certain molecules inhaled from the environment by sending impulses directly to the olfactory bulb of the brain. Humans have about 12 million olfactory receptors, distributed among hundreds of different receptor types that respond to different odors. 12 million seems like a large number of receptors, but compare that to other animals. Rabbits have about 100 million, most dogs have about 1 billion, and bloodhounds, dogs selectively bred for their sense of smell, have about 4 billion. The overall size of the olfactory epithelium also differs between species, with that of bloodhounds, for example, being many times larger than that of humans. Olfactory neurons are bipolar neurons, neurons with two processes extending from the cell body. Each neuron has a single dendrite buried in the olfactory epithelium, and extending from this dendrite are 5 to 20 receptor-laden, hair-like cilia that trap odorant molecules. The sensory receptors on the cilia are proteins, and it is the variations in their amino acid chains that make the receptors sensitive to different odorants. Each olfactory sensory neuron has only one type of receptor on its cilia, and the receptors are specialized to detect specific odorants, so the bipolar neurons themselves are specialized. When an odorant binds with a receptor that recognizes it, the sensory neuron associated with the receptor is stimulated. Olfactory stimulation is the only sensory information that directly reaches the cerebral cortex, whereas other sensations are relayed through the thalamus. Many of the details of the sense of smell were discovered relatively recently by Linda B. Buck and Richard Axel. In 1991, they were the first to pinpoint the olfactory receptors in the back of the nasal cavity of mice. Buck later proposed the way that these receptors could detect so many different smells. Some receptors detect more than one odor, and many odors can be detected by more than one receptor. These detection abilities are applied in different combinations of detection to create a pattern that can both be recognized and remembered. The exact nature of this scent identification, sometimes called odor coding, is still being researched and debated. Buck and Axel later shared the Nobel Prize in Medicine for solving the next piece of the olfactory puzzle. The way that genes code the olfactory receptors, essentially showing the genetic nature of the sense of smell. Illustration A shows a bipolar neuron, which has two dendrites. Illustration B shows a cross-section of a human head. The nostrils lead to the nasal cavity, which sits above the mouth. The olfactory bulb is just above the olfactory epithelium that lines the nasal cavity. Nerve endings, resembling a cluster of small, thin extensions, much like the roots of a plant, run from the bulb into the nasal cavity. Figure 36. 8 in the human olfactory system, A. Bipolar olfactory neurons extend from B, the olfactory epithelium, where olfactory receptors are located, to the olfactory bulb. Credit: modification of work by Patrick J. Lynch, medical illustrator. See Carl Jaffe, MD, cardiologist. Evolution connection. Pheromones. A pheromone is a chemical released by an animal that affects the behavior or physiology of animals of the same species. Pheromonal signals can have profound effects on animals that inhale them, but pheromones apparently are not consciously perceived in the same way as other odors. There are several different types of pheromones, which are released in urine or as glandular secretions. Certain pheromones are attractants to potential mates. Others are repellents to potential competitors of the same sex, and still others play roles in mother-infant attachment. Some pheromones can also influence the timing of puberty, modify reproductive cycles, and even prevent embryonic implantation. While the roles of pheromones in many non-human species are important, pheromones have become less important in human behavior over evolutionary time compared to their importance to organisms with more limited behavioral repertoires. The vomeronasal organ, VNO, or Jacobson's organ, is a tubular, fluid-filled olfactory organ present in many vertebrate animals that sits adjacent to the nasal cavity. It is very sensitive to pheromones and is connected to the nasal cavity by a duct. When molecules dissolve in the mucosa of the nasal cavity, They then enter the VNO, where the pheromone molecules among them bind with specialized pheromone receptors. Upon exposure to pheromones from their own species or others, many animals, including cats, may display the flamen response, shown in Figure 36. 9. A curling of the upper lip that helps pheromone molecules enter the VNO. Pheromonal signals are sent, not to the main olfactory bulb, but to a different neural structure that projects directly to the amygdala. Recall that the amygdala is a brain center important in emotional reactions, such as fear. The pheromonal signal then continues to areas of the hypothalamus that are key to reproductive physiology and behavior. While some scientists assert that the VNO is apparently functionally vestigial in humans, even though there is a similar structure located near human nasal cavities, others are researching it as a possible functional system that may, for example, contribute to synchronization of menstrual cycles in people living in close proximity. Photo shows a tiger snarling. Figure 36. 9. The flamen response in this tiger results in the curling of the upper lip and helps airborne pheromone molecules enter the vomeronasal organ. Credit. Modification of work by Chad/Flicker. Taste. Detecting a taste, gustation, is fairly similar to detecting an odor, olfaction. Given that both taste and smell rely on chemical receptors being stimulated by certain molecules. The primary organ of taste is the taste bud. A taste bud is a cluster of gustatory receptors, taste cells, that are located within the bumps on the tongue called papillae, singular, papilla, illustrated in Figure 3611. There are several structurally distinct papillae. Filiform papillae, which are located across the tongue, are tactile, providing friction that helps the tongue move substances and contain no taste cells. In contrast, fungiform papillae, which are located mainly on the anterior two-thirds of the tongue, each contain 1 to 8 taste buds and also have receptors for pressure and temperature. An illustration shows small, filiform papillae scattered across the front two-thirds of the tongue. Larger circumvallate papillae form an inverted V at the back of the tongue. Medium-sized fungiform papillae are shown scattered across the back two-thirds of the tongue. Foliate papillae form ridges on the back edges of the tongue. A micrograph shows a cross-section of a tongue in which the foliate papillae can be seen as square protrusions about 200 microns across and deep. Figure 36. 10. A. Foliate, circumvallate, and fungiform papillae are located on different regions of the tongue. B. Foliate papillae are prominent protrusions on this light micrograph. Credit A. Modification of work by NCI. Scale bar data from Matt Russell. In addition to those two types of chemically and mechanically sensitive papillae are foliate papillae, leaf-like papillae located in parallel folds along the edges and toward the back of the tongue, as seen in the Figure 36. 10 micrograph. Foliate papillae contain about 1,300 taste buds within their folds. Finally, there are circumvallate papillae, which are wall-like papillae in the shape of an inverted V at the back of the tongue. Each of these papillae is surrounded by a groove and contains about 250 taste buds. Each taste bud's taste cells are replaced every 10 to 14 days. These are elongated cells with hair-like processes called microvilli at the tips that extend into the taste bud pore. Illustrated in Figure 3611. Food molecules, tastants, are dissolved in saliva, and they bind with and stimulate the receptors on the microvilli. The receptors for tastants are located across the outer portion and front of the tongue, outside of the middle area where the filiform papillae are most prominent. The left panel shows the image of a tongue with callouts that show magnified views of different parts of the tongue. The right panel shows a micrograph of the circumvallate papilla, and the bottom right panel shows the structure of a taste bud. At the surface, the taste bud has hair-like projects that are labeled taste hairs. These extend up through a taste pore and lead to a basal cell, gustatory cell, and transitional cell. Figure 36. 11 pores in the tongue allow tastants to enter taste buds in the tongue. Credit. Modification of work by Vincenzo Rizzo. In humans, there are 5 primary tastes, and each taste has only one corresponding type of receptor. Thus, like olfaction, each receptor is specific to its stimulus tastant. Transduction of the 5 tastes happens through different mechanisms that reflect the molecular composition of the tastant. A salty tastant, containing NaCl, provides the sodium ions Na+ that enter the taste neurons and excite them directly. Sour tastants are acids and belong to the thermoreceptor protein family. Binding of an acid or other sour-tasting molecule triggers a change in the ion channel, and these increase hydrogen ion, H+, concentrations in the taste neurons, thus depolarizing them. Sweet, bitter, and umami tastants require a G-protein-coupled receptor. These tastants bind to their respective receptors, thereby exciting the specialized neurons associated with them. Both tasting abilities and sense of smell change with age. In humans, the senses decline dramatically by age 50 and continue to decline. A child may find a food to be too spicy, whereas an elderly person may find the same food to be bland and unappetizing. Link to learning. View this animation that shows how the sense of taste works. Smell and taste in the brain. Olfactory neurons project from the olfactory epithelium to the olfactory bulb as thin, unmyelinated axons. The olfactory bulb is composed of neural clusters called glomeruli, and each glomerulus receives signals from one type of olfactory receptor. So each glomerulus is specific to one odorant. From glomeruli, olfactory signals travel directly to the olfactory cortex and then to the frontal cortex and the thalamus. Recall that this is a different path from most other sensory information, which is sent directly to the thalamus before ending up in the cortex. Olfactory signals also travel directly to the amygdala, thereafter reaching the hypothalamus, thalamus, and frontal cortex. The last structure that olfactory signals directly travel to is a cortical center in the temporal lobe structure, important in spatial, autobiographical, declarative, and episodic memories. Olfaction is finally processed by areas of the brain that deal with memory, emotions, reproduction, and thought. Taste neurons project from taste cells in the tongue, esophagus, and palate to the medulla, in the brainstem. From the medulla, taste signals travel to the thalamus and then to the primary gustatory cortex. Information from different regions of the tongue is segregated in the medulla, thalamus, and cortex. Learning objectives. By the end of this section, you will be able to do the following. Describe the relationship of amplitude and frequency of a sound wave to attributes of sound. Trace the path of sound through the auditory system to the site of transduction of sound. Identify the structures of the vestibular system that respond to gravity. Audition, or hearing, is important to humans and to other animals for many different interactions. It enables an organism to detect and receive information about danger, such as an approaching predator, and to participate in communal exchanges like those concerning territories or mating. On the other hand, although it is physically linked to the auditory system, the vestibular system is not involved in hearing. Instead, an animal's vestibular system detects its own movement, both linear and angular acceleration and deceleration, and balance. Sound. Auditory stimuli are sound waves, which are mechanical, pressure waves that move through a medium, such as air or water. There are no sound waves in a vacuum since there are no air molecules to move in waves. The speed of sound waves differs based on altitude, temperature, and medium, but at sea level and a temperature of 20 OC, 68 OF. Sound waves travel in the air at about 343 meters per second. As is true for all waves, there are four main characteristics of a sound wave: frequency, wavelength, period, and amplitude. Frequency is the number of waves per unit of time, and in sound is heard as pitch. High frequency, greater or equal to 15. DS. For sound frequency, the most commonly used unit is hertz, Hz, or cycles per second. Most humans can perceive sounds with frequencies between 30 and 20,000 Hz. Everyone's ability to hear high frequencies decreases with age. Dogs detect up to about 40,000 Hz, cats 60,000 Hz, bats 100,000 Hz, and dolphins 150,000 Hz, and American shad, Alosa sapidissima, a fish, can hear 180,000 Hz. Those frequencies above the human range are called ultrasound. Amplitude, or the dimension of a wave from the resting position, the equilibrium, to the peak, in sound is heard as volume and is illustrated in Figure 36. 12. The sound waves of louder sounds have greater amplitude than those of softer sounds. For sound, Volume is measured in decibels, dB. The softest sound that a human can hear is the 0 point. Humans speak normally at 60 decibels. A graph shows time going left to right, with amplitude going up and down. The graph shows a regularly repeating sine wave line running from left to right while going gradually up, then down, then up again. The horizontal distance between two crests is the wavelength. The amplitude is the height of the wave from the average height to the peak. On the graph, two waves with the same wavelength are superimposed on one another. The dashed wave does not go as far up or down as the solid wave. Figure 36. 12 for sound waves, wavelength corresponds to pitch. Amplitude of the wave corresponds to volume. The sound wave shown with a dashed line is softer in volume than the sound wave shown with a solid line. Credit, NIH. Reception of sound. In mammals, sound waves are collected by the external, cartilaginous part of the ear called the auricle, then travel through the auditory canal and cause vibration of the thin diaphragm called the tympanum or eardrum, the innermost part of the outer ear, illustrated in Figure 36. 13. Interior to the tympanum is the middle ear. The middle ear holds three small bones called the ossicles, which transfer energy from the moving tympanum to the inner ear. The three ossicles are the malleus, also known as the hammer, the incus, the anvil, and stapes, the stirrup. The aptly named stapes looks very much like a stirrup. The three ossicles are unique to mammals, and each plays a role in hearing. The malleus attaches at 3 points to the interior surface of the tympanic membrane. The incus attaches the malleus to the stapes. In humans, the stapes is not long enough to reach the tympanum. If we did not have the malleus and the incus, then the vibrations of the tympanum would never reach the inner ear. These bones also function to collect force and amplify sounds. The ear ossicles are homologous to bones in a fish mouth. The bones that support gills in fish are thought to be adapted for use in the vertebrate ear over evolutionary time. Many animals, frogs, reptiles, and birds, for example, use the stapes of the middle ear to transmit vibrations to the inner ear. This image shows the structure of the ear with the major parts labeled. The external ear contains the auricle, ear canal, and tympanic membrane. The middle ear contains the ossicles and is connected to the pharynx by the eustachian tube. The inner ear contains the cochlea and vestibule, which are responsible for audition and equilibrium, respectively. Figure 36. 13 sound travels through the outer ear to the middle ear, which is bounded on its exterior by the tympanic membrane. The middle ear contains three bones called ossicles that transfer the sound wave to the oval window, the exterior boundary of the inner ear. The organ of Corti, which is the organ of sound transduction, lies inside the cochlea. Transduction of sound. Vibrating objects, such as vocal cords, create sound waves or pressure waves in the air. When these pressure waves reach the ear transduces this mechanical stimulus, pressure wave, into a nerve impulse electrical signal that the brain perceives as sound. The pressure waves strike the tympanum, causing it to vibrate. The mechanical energy from the moving tympanum transmits the vibrations to the three bones of the middle ear. The stapes transmits the vibrations to a thin diaphragm called the oval window, which is the outermost structure of the inner ear. The structures of the inner ear are found in the labyrinth, a bony, hollow structure that is the most interior portion of the ear. Here, the energy from the sound wave is transferred from the stapes through the flexible oval window and to the fluid of the cochlea. The vibrations of the oval window create pressure waves in the fluid perilymph inside the cochlea. The cochlea is a whorled structure, like the shell of a snail, and it contains receptors for transduction of the mechanical wave into an electrical signal, as illustrated in Figure 36. 14. Inside the cochlea, the basilar membrane is a mechanical analyzer that runs the length of the cochlea, curling toward the cochlea's center. The mechanical properties of the basilar membrane change along its length, such that it is thicker, tauter, and narrower at the outside of the whorl, where the cochlea is largest, and thinner, floppier, and broader toward the apex or center of the whorl, where the cochlea is smallest. Different regions of the basilar membrane vibrate according to the frequency of the sound wave conducted through the fluid in the cochlea. For these reasons, the fluid-filled cochlea detects different wave frequencies, pitches, at different regions of the membrane. When the sound waves in the cochlear fluid contact the basilar membrane, it flexes back and forth in a wave-like fashion. Above the basilar membrane is the tectorial membrane. Visual connection. This diagram shows how sound waves travel through the ear, and each step details the process. A sound wave causes the tympanic membrane to vibrate. This vibration is amplified as it moves across the malleus, incus, and stapes. The amplified vibration is picked up by the oval window, causing pressure waves in the fluid of the scala vestibuli and scala tympani. The complexity of the pressure waves is determined by the changes in amplitude and frequency of the sound waves entering the ear. Figure 36. 14. A sound wave causes the tympanic membrane to vibrate. This vibration is amplified as it moves across the malleus, incus, and stapes. The amplified vibration is picked up by the oval window, causing pressure waves in the fluid of the scala vestibuli and scala tympani. The complexity of the pressure waves is determined by the changes in amplitude and frequency of the sound waves entering the ear. Cochlear implants can restore hearing in people who have a non-functional cochlea. The implant consists of a microphone that picks up sound. A speech processor selects sounds in the range of human speech, and a transmitter converts these sounds to electrical impulses, which are then sent to the auditory nerve. Which of the following types of hearing loss would not be restored by a cochlear implant? Hearing loss resulting from absence or loss of hair cells in the organ of Corti. Hearing loss resulting from an abnormal auditory nerve. Hearing loss resulting from fracture of the cochlea. Hearing loss resulting from damage to bones of the middle ear. The site of transduction is in the organ of Corti, spiral organ. It is composed of hair cells held in place above the basilar membrane like flowers projecting up from soil, with their exposed short, hair-like stereocilia contacting or embedded in the tectorial membrane above them. The inner hair cells are the primary auditory receptors and exist in a single row, numbering approximately 3,500. The stereocilia from inner hair cells extend into small dimples on the tectorial membrane's lower surface. The outer hair cells are arranged in 3 or 4 rows. They number approximately 12,000, and they function to fine-tune incoming sound waves. The longer stereocilia that project from the outer hair cells actually attach to the tectorial membrane. All of the stereocilia are mechanoreceptors, and when bent by vibrations, they respond by opening a gated ion channel. Refer to Figure 3615. As a result, the hair cell membrane is depolarized, and a signal is transmitted to the cochlear nerve. Intensity, volume of sound is determined by how many hair cells at a particular location are stimulated. This diagram shows the structure of the hair cell. The right panel shows a magnified view of the hair cell. The hair cell is a mechanoreceptor with an array of stereocilia emerging from its apical surface. The stereocilia are tethered together by proteins that open ion channels when the array is bent toward the tallest member of their array, and closed when the array is bent toward the shortest member of their array. Figure 36. 15. The hair cell is a mechanoreceptor with an array of stereocilia emerging from its apical surface. The stereocilia are tethered together by proteins that open ion channels when the array is bent toward the tallest member of their array, and closed when the array is bent toward the shortest member of their array. The hair cells are arranged on the basilar membrane in an orderly way. The basilar membrane vibrates in different regions, according to the frequency of the sound waves impinging on it. Likewise, the hair cells that lay above it are most sensitive to a specific frequency of sound waves. Hair cells can respond to a small range of similar frequencies, but they require stimulation of greater intensity to fire at frequencies outside of their optimal range. The difference in response frequency between adjacent inner hair cells is about 0.2%. Compare that to adjacent piano strings, which are about 6% different. Place theory, which is the model for how biologists think pitch detection works in the human ear, states that high-frequency sounds selectively vibrate the basilar membrane of the inner ear near the entrance port, the oval window. Lower frequencies travel farther along the membrane before causing appreciable excitation of the membrane. The basic pitch-determining mechanism is based on the location along the membrane where the hair cells are stimulated. the place theory is the first step toward an understanding of pitch perception. Considering the extreme pitch sensitivity of the human ear, it is thought that there must be some auditory, sharpening mechanism to enhance the pitch resolution. When sound waves produce fluid waves inside the cochlea, the basilar membrane flexes, bending the stereocilia that attach to the tectorial membrane. Their bending results in action potentials in the hair cells, and auditory information travels along the neural endings of the bipolar neurons of the hair cells, collectively, the auditory nerve, to the brain. When the hairs bend, they release an excitatory neurotransmitter at a synapse with a sensory neuron, which then conducts action potentials to the central nervous system. The cochlear branch of the vestibulocochlear cranial nerve sends information on hearing. The auditory system is very refined, and there is some modulation or sharpening built in. The brain can send signals back to the cochlea, resulting in a change of length in the outer hair cells, sharpening or dampening the hair cells' response to certain frequencies. Link to learning. Watch an animation of sound entering the outer ear, moving through the ear structure, stimulating cochlear nerve impulses, and eventually sending signals to the temporal lobe. Higher processing. The inner hair cells are most important for conveying auditory information to the brain. About 90% of the afferent neurons carry information from inner hair cells, with each hair cell synapsing with 10 or so neurons. Outer hair cells connect to only 10% of the afferent neurons, and each afferent neuron innervates many hair cells. The afferent, bipolar neurons that convey auditory information travel from the cochlea to the medulla, through the pons and midbrain in the brainstem, finally reaching the primary auditory cortex in the temporal lobe. Vestibular information. The stimuli associated with the vestibular system are linear acceleration, gravity, and angular acceleration and deceleration. Gravity, acceleration, and deceleration are detected by evaluating the inertia on receptive cells in the vestibular system. Gravity is detected through head position. Angular acceleration and deceleration are expressed through turning or tilting of the head. The vestibular system has some similarities with the auditory system. It utilizes hair cells just like the auditory system, but it excites them in different ways. There are 5 vestibular receptor organs in the inner ear: the utricle, the saccule, and 3 semicircular canals. Together, they make up what's known as the vestibular labyrinth that is shown in Figure 36. 16. The utricle and saccule respond to acceleration in a straight line, such as gravity. The roughly 30,000 hair cells in the utricle and 16,000 hair cells in the saccule lie below a gelatinous layer, with their stereocilia projecting into the gelatin. Embedded in this gelatin are calcium carbonate crystals, like tiny rocks. When the head is tilted, the crystals continue to be pulled straight down by gravity, but the new angle of the head causes the gelatin to shift, thereby bending the stereocilia. The bending of the stereocilia stimulates the neurons, and they signal to the brain that the head is tilted, allowing the maintenance of balance. It is the vestibular branch of the vestibulocochlear cranial nerve that deals with balance. This illustration shows the snail shell-shaped cochlea, which widens into the vestibule. 2 circular organs, the utricle and the saccule, are located in the vestibule. 3 ring-like canals, the horizontal canal, the posterior canal, and the superior canal, extend from the top of the vestibule. Each canal projects in a different direction. Figure 36. 16. The structure of the vestibular labyrinth is shown. Credit: Modification of work by NIH. The fluid-filled semicircular canals are tubular loops set at oblique angles. They are arranged in 3 spatial planes. The base of each canal has a swelling that contains a cluster of hair cells. The hairs project into a gelatinous cap called the cupula and monitor angular acceleration and deceleration from rotation. They would be stimulated by driving your car around a corner, turning your head, or falling forward. 1 canal lies horizontally, while the other 2 lie at about 45-degree angles to the horizontal axis, as illustrated in Figure 36. 16. When the brain processes input from all 3 canals together, it can detect angular acceleration or deceleration in 3 dimensions. When the head turns, the fluid in the canals shifts, thereby bending stereocilia and sending signals to the brain. Upon cessation of accelerating or decelerating, or just moving, the movement of the fluid within the canals slows or stops. For example, imagine holding a glass of water. When moving forward, water may splash backwards onto the hand, and when motion has stopped, water may splash forward onto the fingers. While in motion, the water settles in the glass and does not splash. Note that the canals are not sensitive to velocity itself, but to changes in velocity, so moving forward at 60 mph with your eyes closed would not give the sensation of movement, but suddenly accelerating or braking would stimulate the receptors. Higher processing, hair cells from the utricle, saccule, and semicircular canals also communicate through bipolar neurons to the cochlear nucleus in the medulla. Cochlear neurons send descending projections to the spinal cord and ascending projections to the pons, thalamus, and cerebellum. Connections to the cerebellum are important for coordinated movements. There are also projections to the temporal cortex, which account for feelings of dizziness, projections to autonomic nervous system areas in the brainstem, which account for motion sickness, and projections to the primary somatosensory cortex, which monitors subjective measurements of the external world and self-movement. People with lesions in the vestibular area of the somatosensory cortex see vertical objects in the world as being tilted. Finally, the vestibular signals project to certain optic muscles to coordinate eye and head movements. Link to learning. Click through this interactive tutorial to review the parts of the ear and how they function to process sound. Learning objectives. By the end of this section, you will be able to do the following. Explain how electromagnetic waves differ from sound waves. Trace the path of light through the eye to the point of the optic nerve. Explain tonic activity as it is manifested in photoreceptors in the retina. Vision is the ability to detect light patterns from the outside environment and interpret them into images. Animals are bombarded with sensory information, and the sheer volume of visual information can be problematic. Fortunately, the visual systems of species have evolved to attend to the most important stimuli. The importance of vision to humans is further substantiated by the fact that about 1/3 of the human cerebral cortex is dedicated to analyzing and perceiving visual information. Light. As with auditory stimuli, light travels in waves. The compression waves that compose sound must travel in a medium, a gas, a liquid, or a solid. In contrast, light is composed of electromagnetic waves and needs no medium. Light can travel in a vacuum, figure 36. 17. The behavior of light can be discussed in terms of the behavior of waves and also in terms of the behavior of the fundamental unit of light, a packet of electromagnetic radiation called a photon. A glance at the electromagnetic spectrum shows that visible light for humans is just a small slice of the entire spectrum, which includes radiation that we cannot see as light because it is below the frequency of visible red light and above the frequency of visible violet light. Certain variables are important when discussing perception of light. Wavelength, which varies inversely with frequency, manifests itself as hue. Light at the red end of the visible spectrum has longer wavelengths and is lower frequency, while light at the violet end has shorter wavelengths and is higher frequency. The wavelength of light is expressed in nanometers, nm. 1 nanometer is 1/1000th of a meter. Humans perceive light that ranges between approximately 380 nm and 740 nm. Some other animals, though, can detect wavelengths outside of the human range. For example, bees see near ultraviolet light in order to locate nectar guides on flowers, and some non-avian reptiles sense infrared light, heat that prey gives off. The illustration shows the electromagnetic spectrum, which consists of different wavelengths of electromagnetic radiation. Radio waves have the longest wavelength, about 10 to the third power meters. Wavelength gets increasingly shorter for microwave, infrared, visible, ultraviolet, X-rays, and gamma rays. Gamma rays have a wavelength of about 10 to the 12th power meters. Frequency is inversely proportional to wavelength. Figure 36. 17 in the electromagnetic spectrum, visible light lies between 380 nm and 740 nm. Credit, modification of work by NASA. Wave amplitude is perceived as luminous intensity, or brightness. The standard unit of intensity of light is the candela, which is approximately the luminous intensity of one common candle. Light waves travel 299,792 km per second in a vacuum, and somewhat slower in various media such as air and water, and those waves arrive at the eye as long red, medium green, and short blue waves. What is termed white light is light that is perceived as white by the human eye. This effect is produced by light that stimulates equally the color receptors in the human eye. The apparent color of an object is the color, or colors, that the object reflects. Thus, a red object reflects the red wavelengths in mixed white light and absorbs all other wavelengths of light. Anatomy of the eye. The photoreceptive cells of the eye, where transduction of light to nervous impulses occurs, are located in the retina, shown in Figure 36. 18, on the inner surface of the back of the eye. But light does not impinge on the retina unaltered. It passes through other layers that process it so that it can be interpreted by the retina, Figure 36. 18b. The cornea, the front transparent layer of the eye, and the crystalline lens, a transparent convex structure behind the cornea. Both refract, bend, light to focus the image on the retina. The iris, which is conspicuous as the colored part of the eye, is a circular muscular ring lying between the lens and cornea that regulates the amount of light entering the eye. In conditions of high ambient light, the iris contracts, reducing the size of the pupil at its center. In conditions of low light, the iris relaxes and the pupil enlarges. Visual connection. The left illustration shows a human eye, which is round and filled with vitreous humor. The optic nerve and retinal blood vessels exit the back of the eye. At the front of the eye is the lens with a pupil in the middle. The lens is covered by the iris, which in turn is covered by the cornea, which is a convex bump protruding from the eye. The aqueous humor is a gel-like substance between the cornea and iris. The retina is the lining of the inner eye. A second illustration is a blow-up, which shows that the optic nerve is at the surface of the retina. Beneath the optic nerve is a layer of ganglion cells, and beneath this is a layer of bipolar cells. Both ganglia and bipolar cells are nerve cells with root-like appendages. Beneath the bipolar cell layer are the rods and cones. Rods and cones are similar in structure and column-like. Figure 36. 18a. The human eye is shown in cross-section. B. A blow-up shows the layers of the retina. Which of the following statements about the human eye is false? Rods detect color, while cones detect only shades of gray. When light enters the retina, it passes the ganglion cells and bipolar cells before reaching photoreceptors at the rear of the eye. The iris adjusts the amount of light coming into the eye. The cornea is a protective layer on the front of the eye. The main function of the lens is to focus light on the retina and fovea centralis. The lens is dynamic, focusing and refocusing light as the eye rests on near and far objects in the visual field. The lens is operated by muscles that stretch it flat or allow it to thicken, changing the focal length of light coming through it to focus it sharply on the retina. With age comes the loss of the flexibility of the lens, and a form of farsightedness called presbyopia results. Presbyopia occurs because the image focuses behind the retina. Presbyopia is a deficit similar to a different type of farsightedness called hyperopia, caused by an eyeball that is too short. For both defects, images in the distance are clear, but images nearby are blurry. Myopia, nearsightedness, occurs when an eyeball is elongated and the image focus falls in front of the retina. In this case, images in the distance are blurry, but images nearby are clear. There are two types of photoreceptors in the retina, rods and cones, named for their general appearance as illustrated in Figure 36. 19. Rods are strongly photosensitive and are located in the outer edges of the retina. They detect dim light and are used primarily for peripheral and nighttime vision. Cones are weakly photosensitive and are located near the center of the retina. They respond to bright light, and their primary role is in daytime, color vision. This illustration shows that rods and cones are both long, column-like cells with the nucleus located in the bottom portion. The rod is longer than the cone. The outer segment of the rod contains rhodopsin. The outer segment of the cone contains other photopigments. An oil droplet is located beneath the outer segment of the cone. Figure 36. 19 rods and cones are photoreceptors in the retina. Rods respond in low light and can detect only shades of gray. Cones respond in intense light and are responsible for color vision. Credit: Modification of work by Piotr Sliwa. The fovea is the region in the center back of the eye that is responsible for acute vision. The fovea has a high density of cones. When you bring your gaze to an object to examine it intently in bright light, the eyes orient so that the object's image falls on the fovea. However, when looking at a star in the night sky or other object in dim light, the object can be better viewed by the peripheral vision because it is the rods at the edges of the retina, rather than the cones at the center, that operate better in low light. In humans, cones far outnumber rods in the fovea. Link to learning. Review the anatomical structure of the eye, clicking on each part to practice identification. Transduction of light. The rods and cones are the site of transduction of light to a neural signal. Both rods and cones contain photopigments. In vertebrates, the main photopigment, rhodopsin, has two main parts, figure 36. 20. An opsin, which is a membrane protein in the form of a cluster of alpha helices that span the membrane, and retinal, a molecule that absorbs light. When light hits a photoreceptor, it causes a shape change in the retinal, altering its structure from a bent cis form of the molecule to its linear trans isomer. This isomerization of retinal activates the rhodopsin, starting a cascade of events that ends with the closing of Na+ channels in the membrane of the photoreceptor. Thus, unlike most other sensory neurons, which become depolarized by exposure to a stimulus, visual receptors become hyperpolarized and thus driven away from threshold. Figure 3621. Molecular model A shows the structure of rhodopsin, a transmembrane protein with 7 helices spanning the membrane. A small organic molecule called retinal is tucked inside. B shows the molecular structure of retinal, which has a ring with a hydrocarbon chain attached. A ketone double-bonded oxygen is at the end of the chain. In cis-retinal, the chain is kinked. In trans-retinal, the chain is straight. Figure 36. 20a rhodopsin, the photoreceptor in vertebrates, has two parts, the transmembrane protein opsin and retinal. When light strikes retinal, it changes shape from B a cis to a trans form. The signal is passed to a G protein called transducin, triggering a series of downstream events. Illustration A shows the signal transduction pathway for rhodopsin, which is located in internal membranes at the top of rod cells. When light strikes rhodopsin, a G protein called transducin is activated. Transducin has 3 subunits, alpha, beta, and gamma. Upon activation, GDP on the alpha subunit is replaced with GTP. The subunit dissociates and binds phosphodiesterase. Phosphodiesterase, in turn, converts cGMP to GMP, which closes sodium ion channels. As a result, sodium can no longer enter the cell, and the membrane becomes hyperpolarized. Illustration B shows that the tall, thin rod cell is stacked on top of a bipolar nerve cell. In the dark, the membrane is depolarized, and glutamate is released from the rod cell to the axon terminal of the bipolar cell. In the light, no glutamate is released. Figure 36. 21. When light strikes rhodopsin, the G protein transducin is activated, which in turn activates phosphodiesterase. Phosphodiesterase converts cGMP to GMP, thereby closing sodium channels. As a result, the membrane becomes hyperpolarized. The hyperpolarized membrane does not release glutamate to the bipolar cell. Trichromatic coating. There are 3 types of cones with different photopsins, and they differ in the wavelength to which they are most responsive, as shown in Figure 36. 22. Some cones are maximally responsive to short light waves of 420 nm, so they are called S cones, S for short. Others respond maximally to waves of 530 nm, M cones, for medium. A third group responds maximally to light of longer wavelengths, at 560 nm, L, or long cones. With only one type of cone, color vision would not be possible, and a 2-cone dichromatic system has limitations. Primates use a 3-cone trichromatic system, resulting in full color vision. The color we perceive is a result of the ratio of activity of our 3 types of cones. The colors of the visual spectrum, running from long wavelength light to short, are red 700 nm, orange 600 nm, yellow 565 nm, green 497 nm, blue 470 nm, indigo 450 nm, and violet 425 nm. Humans have very sensitive perception of color and can distinguish about 500 levels of brightness, 200 different hues, and 20 steps of saturation, or about 2 million distinct colors. Graph plots normalized absorbance for rods and S, M, and L cones against wavelength. For all 4 cell types, the trend is an approximately bell-shaped curve with a steeper decrease than increase. For S cones, the peak absorbance is 420 nanometers. For rods, the peak absorbance is 498 nanometers. For M cones, the peak absorbance is 534 nanometers. For L cones, the peak absorbance is 564 nanometers. Figure 36. 22 human rod cells and the different types of cone cells each have an optimal wavelength. However, there is considerable overlap in the wavelengths of light detected. Retinal processing. Visual signals leave the cones and rods, travel to the bipolar cells, and then to ganglion cells. A large degree of processing of visual information occurs in the retina itself before visual information is sent to the brain. Photoreceptors in the retina continuously undergo tonic activity. That is, they are always slightly active even when not stimulated by light. In neurons that exhibit tonic activity, the absence of stimuli maintains a firing rate at a baseline, while some stimuli increase firing rate from the baseline and other stimuli decrease firing rate. In the absence of light, the bipolar neurons that connect rods and cones to ganglion cells are continuously and actively inhibited by the rods and cones. Exposure of the retina to light hyperpolarizes the rods and cones and removes their inhibition of bipolar cells. The now active bipolar cells in turn stimulate the ganglion cells, which send action potentials along their axons, which leave the eye as the optic nerve. Thus, the visual system relies on change in retinal activity, rather than the absence or presence of activity, to encode visual signals for the brain. Sometimes horizontal cells carry signals from one rod or cone to other photoreceptors and to several bipolar cells. When a rod or cone stimulates a horizontal cell, the horizontal cell inhibits more distant photoreceptors and bipolar cells, creating lateral inhibition. This inhibition sharpens edges and enhances contrast in the images by making regions receiving light appear lighter and dark surroundings appear darker. Amacrine cells can distribute information from one bipolar cell to many ganglion cells. You can demonstrate this using an easy demonstration to trick your retina and brain about the colors you are observing in your visual field. Look fixedly at Figure 36. 23 for about 45 seconds. Then quickly shift your gaze to a sheet of blank white paper or a white wall. You should see an afterimage of the Norwegian flag in its correct colors. At this point, close your eyes for a moment, then reopen them, looking again at the white paper or wall. The afterimage of the flag should continue to appear as red, white, and blue. What causes this? According to an explanation called opponent process theory, as you gazed fixedly at the green, black, and yellow flag, your retinal ganglion cells that respond positively to green, black, and yellow increase their firing dramatically. When you shifted your gaze to the neutral white ground, these ganglion cells abruptly decreased their activity, and the brain interpreted this abrupt downshift as if the ganglion cells were responding now to their opponent colors, red, white, and blue, respectively, in the visual field. Once the ganglion cells return to their baseline activity state, the false perception of color will disappear. A Norwegian flag is shown in false colors of green, yellow, and black. Normally, the colors are red, white, and blue, like the American flag. Figure 36. 23 View this flag to understand how retinal processing works. Stare at the center of the flag, indicated by the white dot, for 45 seconds, and then quickly look at a white background, noticing how colors appear. Higher processing. The myelinated axons of ganglion cells make up the optic nerves. Within the nerves, different axons carry different qualities of the visual signal. Some axons constitute the magnocellular, big cell, pathway, which carries information about form, movement, depth, and differences in brightness. Other axons constitute the parvocellular, small cell pathway, which carries information on color and fine detail. Some visual information projects directly back into the brain, while other information crosses to the opposite side of the brain. This crossing of optical pathways produces the distinctive optic chiasma, Greek, for crossing, found at the base of the brain and allows us to coordinate information from both eyes. Once in the brain, visual information is processed in several places, and its roots reflect the complexity and importance of visual information to humans and other animals. One route takes the signals to the thalamus, which serves as the routing station for all incoming sensory impulses except olfaction. In the thalamus, the magnocellular and parvocellular distinctions remain intact, and there are different layers of the thalamus dedicated to each. When visual signals leave the thalamus, they travel to the primary visual cortex at the rear of the brain. From the visual cortex, the visual signals travel in two directions. One stream that projects to the parietal lobe in the side of the brain carries magnocellular, where, information. A second stream projects to the temporal lobe and carries both magnocellular, where, and parvocellular, what, information. Another important visual route is a pathway from the retina to the superior colliculus in the midbrain, where eye movements are coordinated and integrated with auditory information. Finally, there is the pathway from the retina to the suprachiasmatic nucleus, SCN, of the hypothalamus. The SCN is a cluster of cells that is considered to be the body's internal clock, which controls our circadian, day-long cycle. The SCN sends information to the pineal gland, which is important in sleep-wake patterns and annual cycles. Link to learning. View this presentation to review what you have learned about how vision functions.